Today I will talk about rtPCR, reverse transcription polymerase chain reaction. This term has been very popular nowadays, as you know, because of the COVID-19 situation. The SARS-CoV-2 virus that poses in plus strand RNA, and that is being detected through this technique, rtPCR. We will learn about that today. So, since it is a reverse transcription process, it starts with a mRNA, and via reverse transcription, the mRNA is converted into cDNA, complementary DNA. That's why the term is reverse transcription, followed by polymerase chain reaction. So first, an RNA molecule will be converted into a cDNA molecule, and cDNA molecule will be further be amplified through PCR. So the RNA molecules of all eukaryotic system have a poly A tail at the 3' end, and that's make it relatively easy to perform this reverse transcription process. So if you design, one of your primer could be oligo DT. So if you design, oligo DT will bind with the poly A tail, so this will act as your primer. So if you add the reverse transcriptase enzyme, reverse transcriptase plus all the D NTPs, then what will happen? This oligo DT, it will copy this, it will use this mRNA as a template, and it will synthesize a DNA strand over there. That will be the first strand cDNA. So let's see how it happens. So here is your mRNA, 3' end, and this was the, suppose I am drawing this like a box like this. This is prime. Oligo DT. This is oligo DT primer. Okay. So the RT, reverse transcriptase enzyme, it will use this 3' hydroxyl end and will incorporate nucleotide at this 3' end, and will extend this, will synthesize a new DNA. This is called the first strand cDNA. So this is the first strand cDNA. So it is called first strand cDNA synthesis. After this, since it is an RNA DNA hybrid, the second strand cDNA has to take place. So this mRNA has to be removed. So it is removed by two processes. One of the processes is using RNase H enzyme. Another is you can remove this RNA via alkali treatment. So sodium hydroxide treatment. RNAs are labile to alkaline environments, so they will degrade in alkaline environments. But RNase H, it is recommended that this first process, that is using RNase H, is better. RNase H, it is an enzyme. It acts on the RNA DNA hybrid, and it clips the RNA only specifically. It does not affect the DNA. So suppose you are using RNase H, sorry. So what will happen? This RNA molecule, it will get degraded into fragments like this. Okay, and this is your DNA, oligo DT primer, and this was your DNA, first strand cDNA. So now your RNA has been clipped into fragments like this. There are poly A tails also at this end. So everything is clipped now, and all these fragments have 5', 3', 5', 3' ends. So if you use DNA pol 1, DNA pol 1 will bind at these positions. It will use these fragmented RNA as primers, and just like a nick translation process, it will remove one by one the nucleotides of the RNA fragment that is coming in front of its path. In its path, it will remove via 5' to 3' exonuclease activity, and it will add new DNA on the 3' ends at the same time. So it will ultimately replace this mRNA fragments with a DNA molecule. This part will also be replaced by the DNA molecule. Up to this. So this is the second strand cDNA that has been synthesized. This is called second strand cDNA synthesis. So now your double-stranded cDNA is ready, and you can go for polymerase chain reaction, provided you should be knowing some part of the sequence of this region so that you can design another primer, because PCR requires a pair of primers. So this is how reverse transcription PCR works. Reverse transcription PCR has many applications. One of the applications is disease detection. The RNA viral diseases can be detected via this method. There are other uses like you can measure the expression level of certain gene. You can design cDNA libraries. You can make cDNA library by this method, by following this method. And again, you can also use it for synthesizing what is called ESTs, expressed sequence tags. Those are required for genomic research. We will discuss about these things later on in application part. So this was about reverse transcription PCR. Thanks for watching.